Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 142nd seminar, and I call it on MIT Day. This is a rare week when I am in San Diego, although I officially started my new position as a full professor at J. Craig Venter Institute, San Diego, in May this year. I have been traveling all over the world to give invited talks and discuss the solutions to global problems through collaborations between my NSF Global Center and other research groups or centers. As a result, I have seldom stayed in San Diego, but starting this week, there are many conferences in San Diego, including AICG CRISPR conference I just attended, also gave a talk earlier this week, and of course, AICHE annual meeting this month, very soon. I'm so excited to host some colleagues and also a Cell Journal editor at JCVI, my institute, for a mini symposium and seminar this month, next week, and 2 weeks from now, and see many of you soon in San Diego. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Robert or Bob Langer at MIT. He does not need any introduction because we all know him, or introducing him briefly would be almost impossible. I will try. I would say he is the most influential and famous engineer alive in this era, with more than 100 companies and numerous patents utilized for real-world applications, including the core technology used for the COVID vaccine development and many other drug delivery and tissue engineering technologies. He is a role model for many people, including myself, especially due to the challenges he experienced and overcame when he was young or assistant professor, because his innovative work was not recognized by petroleum-oriented senior chemical engineers and professors. I personally had a huge honor to receive the Langer Prize for Innovation and Entrepreneurial Excellence in 2022 and formed a startup company called Moonshot Bio thanks to this award. Receiving this award was one of the 6 best things that happened to me following my miraculous recovery from dying years and exodus from my previous institute, Washington University in St. Louis, meeting my wife and marriage to her, my daughter's birth, and my admission to MIT as a graduate student. Okay, the introduction was longer than I planned or expected. Professor Langer, thank you so much for your amazing contribution to the society and education, as well as being a role model, and the virtual podium is all yours now. And thank you again.
Well, thank you so much for having me. I thought I would briefly tell you about some of the things in my career. So I am a chemical engineer, as was mentioned, but when I finished my degree in 1974, pretty much all my friends went to work for oil companies, and I wasn't that excited about that. I kept looking for a way that I could use my chemical engineering skills to help people. I tried to get teaching jobs and no one would hire me. And then I had this idea about maybe working in medicine. And at that time, it was very rare for a chemical engineer to do that, especially experimental medicine. anyhow, I wrote to a lot of hospitals and medical schools, and again, nobody would hire me or even write me back. But finally, one day, one of my friends said to write to a surgeon named Judah Folkman, and he hired me. And he had this idea that if you could stop blood vessels, you could stop cancer. And that to me was very exciting. It didn't pay like the oil companies, but I thought it was something that was really important. The problem that we had in terms of doing this is that there wasn't a good way to study blood vessels. So we had to invent one. And what we did is shown on the first slide, which you could see on the upper left, is there's certain places in the body, chick chorioallantoic, or that exist, like chick chorioallantoic membranes or corneas. And here you see sort of a schematic of a cornea. Our idea was to put tumors in, and then I had isolated different large molecules like proteins, glycoproteins, and others. And our idea was to put a tumor in, and then next to it, a tiny little delivery system like a nanoparticle or microparticle that could deliver these large molecules. Now, this was 1974. That had never been done before. There was only one company in the world at this time, Alza in California, working on advanced drug delivery systems. Dr. Folkman was on their board. He went out and asked them, could they help? But they all told him what you see on the second slide, which is in the upper right, that the molecules were too big to get out, just like we're all too big to walk through walls. At any rate, I spent several years working on this, and I found over 200 different ways to get it to not work. But eventually, we were able to publish a paper in Nature showing, which you see on the third slide, that's the first one on the bottom, that left, that you could, with certain techniques, release large molecules. It's also the very first paper that ever showed that you could release and deliver nucleic acids. And for reference, RNA and DNA are nucleic acids. Yet, when I first started lecturing on this, it was ridiculed. Many scientists came up to me after a talk I gave, and I was only 27 years old at the time, and they said, well, this has to be wrong. They said, again, as you look on the fourth slide, they said this can't work because the molecules were too big. Also, they said the solvents we were using would destroy whatever we put in. But I kept at it. But nonetheless, what happened was, I couldn't get research grants. My first 9 were rejected. I couldn't get a faculty job in chemical engineering. No one would hire me. They all thought this bio stuff I was doing didn't make any sense. I finally got a job in a nutrition department. I think the reason I got it was because the head of that department was a benevolent dictator kind of department head. And he liked me, so he offered me a job, but he didn't ask anybody else in the department what they thought, which would have been okay, except that the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, on this first slide now, on this, which is the 5th slide, this is coming from Michael Marletta, a very close friend of mine, who later became head of Berkeley's chemistry department. And he's just talking about what it was like to be with me at MIT in 1979. And I'll read you part of what he said. He said, one evening, he said, I went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. Nonetheless, I'm very stubborn. I kept at it. We had published this in Nature and we advanced it. And then we were able to use it to actually isolate the first blood vessel inhibitors with that assay that I mentioned. We published a paper in Science in 1974 on that. And what that would ultimately lead to, though it took 28 years and more, is to what you see here. That starting in 2004, you see one after the next blood vessel inhibitors approved by FDA. Avastin, the first of these, is probably the number 2 or 3 best-selling biotech drug in history. And they're not only approved for different cancers, all kinds of cancers, but they're also the only drugs that can be used for treating blindness in the back of the eye. Drugs like Eylea and Genentech to treat diseases like macular degeneration and diabetic retinopathy. So that was very exciting to see that work by Genentech and others finally make an impact, building on what we'd done. At any rate, going back to the 70s, we can just, going back to the 70s, 1970s, Dr. Folkman and I decided to file a patent, which was very unusual for Children's Hospital at the time. They never had a patent before. We filed one for the first time, and 5 years in a row it was rejected. And the head of the hospital came to see me and said, Bob, you have to quit. You're wasting so much money. But I don't like to quit. So I started to think, how could we convince the patent examiner to allow this patent, legally, of course? And science wasn't working. But as I told you in the beginning, when we first started doing this, everyone said it was impossible. It could never work. I wondered, maybe people had written that down. So in 1982, I did a science citation search looking back at who referred to our Nature paper. And here is one paper I found. I had no idea it was written, but you can just look at it. But what's really important is it talks about what we did. And the last sentence says, however, Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So we showed that to the patent examiner, and he said, gee, he had no idea, but he said, he'd tell you what, he will allow this patent if I could get written affidavits from each of these 5 people that they really wrote that. So I wrote them. They were nice enough to write me they really wrote it. And on the next slide, we got this incredibly broad patent. And I thought, gee, we had all this, and, large companies would use these things. And we actually got 2 arrangements from multi-billion-dollar pharmaceutical and animal health companies. And I was so excited about that. But what happened was, this was the early 80s, they worked on it for maybe 6 months to a year, and they gave up. So Alex Klebanoff, another friend of mine, said, Bob, we should start our own company. So we did. We started a company, Enzatech, with 4 of my students. And over the years, we've started some 40 companies. If we go to the last slide, I just want to mention many of the products that have come out from that, all kinds of ways of, we can maybe move that up, of delivering molecules to treat everything from opioid addiction to heart disease, like you see a drug-eluting stent, to the COVID vaccines with Moderna, a company I started with 3 others, and ways to treat schizophrenia. It's not limited, by the way, to just humans. On the bottom right, Yanni Zohar and I came up with the idea of using these for aquaculture, and it helped make that a $250 billion industry. And then, of course, the other area that we've helped get started is this area of tissue engineering, which could make new tissues and organs from scratch, and maybe organs and tissues on a chip. So I want to thank you very much for inviting me. It's been an honor to present all this to you, and I wish you all the best of luck. Thank you so very much.
Thank you so much. And that story I heard a couple of times, and people, especially young people, only see the initial point and the final point. I mean, your reputation. But I believe everyone needs to remember, to be that, pioneer, you need to go through very difficult time. Once you overcome, you will be, recognized. And then that part I need to emphasize one more time. That is actually a message we wanted to deliver. And then another thing I want to add, just dream high, never give up, and then you will contribute to the society. That is a brilliant experience and a journey. I really thank you for never give up and then make this world much better place than 30 years ago or 40 years ago. Thank you.
Well, thank you, and thank you for what you're doing. It's been an honor to present, and wish you all the best of luck. Thank you so much.
Thank you.
That
Bye-bye.
was really fascinating, actually. So the message was persistency, like consistency over excitement. That was really fascinating, the story. Thank you very much for sharing that with us.
Thank you. Thank you as well. Thank you all.
Sure.
Bye-bye.
Okay, now the main speaker of today, Dr. Leili Rohani is a research scientist at MIT in the lab of Ron Weiss, another SynBYSS pioneer speaker. She was a stem cell scientist at the School of Biomedical Engineering, University of British Columbia. Her research has been focused on stem cell, regenerative medicine, cell therapy, cell fate engineering, with the intent to provide a platform for future gene and precision therapies for heart disease. She is passionate about combining tissue engineering, single nuclei RNA sequencing, and synthetic biology tools to create a human single-cell atlas of heart disease as a basis for understanding, diagnosing, and monitoring, and also treating heart disease. Our end goal is to look at the SynBio platform, such as tissue engineering, single nuclei, RNA-seq, and synthetic biology as a new vocabulary for disease studies to determine the ways in which cells and disease genes act, which cells are disrupted in disease, which also programs change in them, what mechanisms underline their dysregulation, how their cell-cell communication are affected, and what would be the impact of therapies. Beyond her research, she's passionate about science communication, networking, collaboration. Leili, thanks so much for your time today. Please take it away. Thank you.
Thank you very much for a nice introduction, and thanks for inviting me, having me here. And actually, I wanted to tell a special thanks for all what you do for the community. You are now a YouTube superstar with collection of scientists, junior scientists, senior scientists, and synthetic biology. So it's really amazing. Also, the opportunity that you give to like the young scientists like myself to be more exposed to the community, having more exposure. That's fantastic. So thanks for all what you do.
My pleasure.
Yeah.
Go ahead.
So let me share my screen and then can everyone, give me a second, stop sharing. Just going back to the first slides to make sure that we have all set here. All right. So everyone can see my slides. I don't know why it changed when I go to the presentation. So great. So it's all clear, so everyone can see my
Yes, go ahead,
slides.
please.
Okay, great. Okay. So this is the topic that I've chose for my presentation today: synthetic biology, you and me, and how we can apply synthetic biology to combat disease. So when I start presenting my research, I start talking about my science journey. I started my science journey on stem cell science and stem cell biology from Royan Institute back in Iran. Then I moved to Germany to do my PhD at the Fraunhofer Institute. And as part of that, I did knowledge translation. I went to Vienna, worked with a Versailles company to transfer microRNA, mRNA-based cellular reprogramming to Versailles GmbH in Vienna. And then right before coming to MIT, I was in Canada, School of Biomedical Engineering in Calgary and UBC. And for my final destination so far has been MIT Synthetic Biology Center, which I guess it's been so far an amazing situation for me, and I guess it was a great decision because it's very much tied to my passion to apply synthetic biology to combat disease. So I've been working on many different contexts during my research journey, but I mean, so if I want to give a summary about my research and what I have done so far, I've been working mostly on cell fate engineering, as you mentioned, and cell therapy and regenerative medicine. So, and I've been planning always to change the fate of the cells with the goal to have better cell types for cell therapy. So I have used many different tools so far, but I'll just briefly give introduction about a few of them, and then more extensively, I will explain about two of those tools. So one of the tools that I have used so far for changing the fate of the cells been bioreactors, shear stress that we apply mechanotransduction to change the fate of the cells, stem cells from prime to naive human pluripotent stem cells, and that's where we got invited to. I was invited to Stem Cell Podcast to talk about this research. And then the other tool that I have used so far for changing the fate of the cells has been mRNA molecule back in Germany. We applied it because we wanted to provide a very translational approach to generate more clinical-based mRNA iPS cells. So my motivation to engage in synthetic biology research coming from the two last research I have done so far, one before coming here to MIT, and then the current one that I am applying that I'm working on at MIT extensively. So for the type of synthetic biology we applied for this, for the first one, we applied combinatorial screening design and fine-tuning of liver organoid, iPS-derived liver organoid toward EpCAM progenitor. So we came up with a specific math modeling to change the fate of the cells toward a specific cell type in liver organoid. And I will explain later why we wanted to do this. And the second part is decoding cardiac fibrosis and activated solid states, progressive cardiac fibrosis and activated solid states using hybrid synthetic promoter and microRNA cell-based classifier. It's a gene therapy-based approach platform that we want to bring to, if possible, we want to bring it to the market to sell it as a platform to tackle heart disease. So I will focus first on the combinatorial screening for fine-tuning of iPS-derived liver organoid. So as I mentioned, we came up with a specific math modeling, design of experiments, and a combination of computational biology and math modeling, and small molecules for hepatocyte signaling to change the fate of liver organoids toward a specific cell type, to enrich EpCAM progenitors, which are stem cell types in liver organoid. And it was in collaboration, in a great collaboration back at the University of Calgary School of Biomedical Engineering with Mark and Derek. So it was really successful collaboration. One of those very good collaboration that shows like how interdisciplinary research worked pretty well, because I had a stem cell background, they had engineering background, so it worked pretty well. It was a very nice combination. So why we wanted to apply this design of experiments and computational modeling to tune a specific, to tune liver organoid toward a specific cell types, because it came from two specific bottlenecks. First, batch-to-batch variation in cell population within organoids, which is a universal problem in all organoids field actually. So we have different cell type or different cell population, at least different percentage of them batch-to-batch. And the second one, the scalable and high throughput systems for scalable generation of liver organoids, so that they can keep all cell population there. The idea was that we wanted to apply multiple factors contributing to stem cell fate decision. So we wanted to combine different factors to add them to liver organoids at the same time. So it's like simultaneously to save time and also to get into correlation effect. Because most of the biologists apply different small molecules to tune a specific cell types in liver organoids or stem cell, applying on stem cells, but that's not something that efficient, because we know that those factors have correlation effect on each other as well. So it's not just one effect. So that's something that we wanted to, that's something that we wanted to, we came up with the idea to apply design of experiments and math modeling to liver organoid. So a brief overview of how we engineered this liver organoid. We started with iPS cells, human embryonic stem cells. We applied them to microval, AgriVal, which was patented and commercialized by Mark Green, the engineer, my very own collaborator back at the University of Calgary Bioengineering School. So we generate those spheres in AgriVal, and then we follow 3 different stages of differentiation for this liver organoid. Stage 1, definitive endoderm, stage 2, hepatobolus, and stage 3, maturation. And we kept them all on shaker for 2-3 weeks to become a liver organoid. So, and then we also tried this approach with the other platform, just to show that this is reproducible and applicable to other platforms. So far, it was good. So we expected to see liver organoids batch to batch, not like at scale. So how we could get, like, I mean, if we wanted to use it for drug screening, that was the main goal. And so we assessed them with different functional tests for liver organoid. It was fine. They showed all the typical markers of liver organoid. But when we exposed them to drug, they couldn't break the drugs into their metabolites. So and then we tried different things. We were involved in that, testing that procedure for at least 6 months to be able to make them to make them more functional to break the drugs into their metabolites. But that never happened. So when we look at, but something that came up, so science actually told the results and the cells were the boss, and they tuned us to a different direction, which was fantastic actually. So when Doug and I started getting this organoid in Agriwell, we noticed that we start with homogeneous population. So the starting point was a homogeneous population, but then towards the end of differentiation, so the organoids, at least based on the morphology, they change. So at least we were able to capture 3 different phenotypes, like disorganized clusters, big open types, like the cystic types, and small dark ones. So we came to the idea that maybe this organoid, because we started a specific population with homogeneous population, so it's exactly identical. But when we go to the end, they change. So maybe they talk to each other, and that's it, like they communicate with each other. So it's not like that there is some competition or something there that changed this homogeneity toward more heterogeneity. So either physical interaction or chemical interaction. So that's why we decided to classify them and then screening them every single day, at least based on their morphology and phase contrast microscopy, to see if we can simply classify them into different subtypes. So, and actually, the interesting part was that we were able, although we started everything from the beginning, all the protocols, all those organoids experienced the same protocol, the same treatment, but they've changed differently from the starting point to the end. And that was not something relevant to chemical, to like what we added to the cells. It was relevant to what they communicated to each other. And based on what based on this modeling, this actually characterizing them or checking them every day, we found, like Puyan, actually one of the collaborators back at the University of Calgary School of Biomedical Engineering. So we found a specific model, an algorithm there, so for these organoids. So we noticed that we start with more basically more flower-shaped one. We start with more like a brightness toward the core, having the core like the very hard core in the middle. So that means that this communication, like physical or chemical interaction, tuned them toward a specific subpopulation of liver organoids. So they were all liver organoid, but different subpopulation. So interestingly, we were able to isolate them or separate this liver organoid using a FICO. So like based on the density gradient, which was a simple idea. We just wanted to see how we can characterize deeply this subtype of organoid, which they are still hepatic organoid, but different subtypes within the culture. So we applied density gradient FICO. Isolate them by FICO, and we isolate them using density gradient, and we ended up with having 3 different subpopulations, like top cystic, middle flower shape, and bottom round shape, and that's why we called them, just based on the morphology. And the next step, we wanted to see if they really how is the screening, how we screen them, how is the gene expression in these subtypes. So very interesting, the subtype population, cystic one, showed the expression of markers relevant to stem cell, like liver stem cell, like CFTR, EpCAM, cytochrome 7 and 17, and LGR5. So it was not like that they were differently morphologically or based on density gradient. They were different actually based on the gene expression. So they were really different, a subtype of hepatocyte actually. So, and that's why we found that, okay, so we really have heterogeneous population like other organoid system, but how we can make them more unique population, either more hepatocyte or EpCAM progenitors, to be able to screen them for a specific application. So we came up with a specific math model because I work with the engineers and we were interested to apply math and math modeling to this, number one. Number two, design of experiments is a very good approach for fate tuning because we were able to apply different factors at the same time. We started with this specific design, differentiative screening design, to enrich a specific subpopulation in liver organisms. And we were not able, we didn't know at that time which population. It was like totally blind. And the design was 46 factor designed, and we screened 29 factors. the 29 factors which were shown, which were promote hepatic differentiation according to the literature. And we came up with 3 different levels. So 29 factors and 3 different level dose for each factor. And each factor had a unique combination of these different small molecules. And of course, robot was something that made life easier for us. So we noticed that when we apply these molecules to this organate, the molecules that we got from the literature, so 6 of them, 6 of those small molecules showed significant difference, and they showed like they changed the fate of organoids toward different types. So this is the original media that we applied, and then the second media that we applied with using these 6 small molecules that show difference to tune the liver organate fate, they became cystic. So what we did, we came up with the second design, central composite design. We got these 3 factors, made a new design, but this time more fine-tuning. It's like that we started from high throughput screening toward more fine-tuned dynamic system to be able to tune cells within liver organelle. So this second design was central composite design with 5 different levels. And using this system, well, we were able to come with a model, basically a dynamic, let's say the dynamic model, or fine-tune a dynamic model to fine-tune cell types within organelles, depends on the application. So at some point, we were able to get some of the CFTR high expression and CYP2C19 low expression, and some of this graph. So basically what I'm trying to say is that, so we had a dynamic spot here. So right before here or here, we were able to change the fate of those organoids based on this model to be hepatocyte or to be EpCAM progenitor. So we made a dynamic model, which was great. And then we analyzed these organoids using single-cell RNA sequencing, which I don't want to go to that now because I'm just trying to keep the track of time. But we had tons of data characterizing this organoid, how we were able to change this organoid to our specific cell type, to our EpCAM progenitors, using single-cell RNA sequencing. So the stories are there, and the manuscript is on the preparation still. And the second topic that I'm very keen, and I'm doing now right at the MIT Synthetic Biology Center in collaboration with the people at Ron's lab, is that we want to apply synthetic biology to combat disease. So basically, if possible, using programmable microRNA input-output, using genetic circuits designed for microRNA input, microRNA output, so multi-input logic, and then seq-RNA as a backup. And we are lucky here because microRNA got a Nobel Prize for Physiology and Medicine a few days ago. So it seems that we are on the right path, and they also, even in the culture, in the environment, in the real environment, in the lab, we really get the results, which are very promising and aligned with what we want. So a little bit story, brief story about this work, how to apply synthetic biology to combat disease. So I will start with the core team that extensively helping me on this. And in Ron's lab and outside external collaborators back in UBC, in Germany, and Washington, and even in Boston University. So the whole idea is that we bring a hybrid platform, a translational platform for precision target therapy for heart disease. So it's basically gene therapy. And it consists of 3 different tools, starting with single nuclear RNA sequencing to synthetic biology. So tool 1 is single-cell RNA nuclei RNA sequencing of patient heart tissues. And these are the patients who underwent heart transplantation. Speaking about patient, this is the disease, the disease that we apply and we work on, that is hypertrophic cardiomyopathy. So basically COVID forced us to get into this research, because we had to wait for a platform for a long time in the UBC. And we had to do some research assessment because of the engineering heart tissue platform across the border. So we had to do some resource assessment to get into biobank to see how we can do something as a site, which is now my main project, what we can do as a site to be able to cover the time that we're waiting for the platform across the border. And that's how we came up with this story. The single nuclear, we have tool one, that, like what I said, the platform, a translational platform consists of 3 different tools. Tool one, single nuclear RNA sequencing from patient heart tissues. And these are the patients who underwent heart transplantation. They donated heart tissue to the biobank. Tool two is iPS-derived organoids, cardiac organoids, either from the same patient or other iPS cell lines that have hypertrophic cardiomyopathy. And then tool three is synthetic biology, which is something that we are very focusing in the Rand lab. So we want, and which synthetic biology tools? We want to apply genetic GRN engineering, transcription factor perturb in collaboration with Germany. Synthetic promoter spec library that we already have the tools in the lab, and also we have the publication on that, and mRNA-based cell-based classifier. So basically mRNA are sensors and transcription factor are the real target. That's what we do at the moment. And of course, this is not possible without without the help from external collaborators, the UBC, Germany, U Washington. So what is the innovation here? The innovation is that we want to apply, and why we want to apply synthetic biology to this disease is because it's a very heterogeneous disease condition, and it comes up with a dynamic system. So it's like that the mutation-driven SynBYSS HCM, they always go through dynamic system. So it's like that, it's a very heterogeneous system. So it's not like that we can target easily by CRISPR, which is a conventional method to target or reverse the disease phenotype. And the whole novelty coming from the novel core transcription factors that we are into, and we have those results in single-cell RNA sequencing. And those transcription factors drive fibrotic feature in cardiomyocyte populations, and also they derive inflammatory biomarkers or derive inflammatory features in cardiomyocyte. That's why we think that secondary part of this fibrotic would be cardioimmuno or cardiometabolism, which we are into that. And then we want to have, like what I mentioned earlier in the earlier slides, we want to apply gene regulatory network in the context of disease, manipulate GRN to reverse disease phenotype. So basically, the hybrid platform on synthetic biology part consists of an enhanced cell-state specificity-based circuits, so basic synthetic promoter, multi-input microRNA cell-based classifier, which our lab has very long background on this, like history on this. So Ron is one of the pioneers on microRNA cell-based classifier to detect the cancer. Now we want to apply it we want to bring it to the context of heart disease and other disease in the lab. And then all comes from gene regulatory network inference, like the computational model and computational data that we have when we want to apply it in vitro, validated in vitro. And the end goal is that we can selectively silence and activate at precise time, dose, and targets in specific types and state. So we can, at the high level, we can detect if they are cardiomyocyte, if they are not cardiomyocyte, and the second level, if they are fibrotic versus non-fibrotic. So cell type, cell state, and also right time and right dose. And these are all down the roads, and we're working on that. So speaking about the first tool and the single nuclear RNA-seq analysis, we want to start from, and like what I mentioned earlier, we want to start from in silico to go to in vitro modeling. And the two main questions here is that two main questions here are, what are the main cell population affected by disease? Like heterogeneity within one cell types, like cardiomyocyte. So we know that cardiomyocyte is based on the data, a specific population that affected by disease, and how they can communicate these mutated cells, how they can communicate to the neighboring cells toward extreme fibrosis, which is a major phenotype of disease. And the manuscript is in preparation with the colleague at the UBC, ex-colleague at UBC. So we have patient of cohort, hypertrophic cardiomyopathy and control. And like what I mentioned, the phenotype is extreme fibrosis, but we think that progressive fibrosis start way earlier that we see the phenotype. And why we came up with this story. So I will pass quickly these slides because I just want to focus more on the real results and the real story, the novel part of that, which was basically checking the regulatory part in this using the single-cell RNA sequencing and the regulatory part in this cardiomyocyte population. And based on this gene regulatory network analysis, we know that this hypertrophic cardiomyopathy and the cardiomyocyte population having a specific transcription factor expression of some of the transcription factor and regulons, which drive cardiac fibrosis way earlier that this disease goes to fibrosis. That's why we have a hypothesis that, and they are dysfunctional cardiomyocyte actually, and that's why we are into hypothesis that something upstream, like gene regulatory network and upstream, sending signal to this cardiomyocyte population, and they are the major driver to send signal to the neighboring cells, like fibroblasts, endothelial cells, toward extreme fibrosis. And that is something that now we are doing in the lab. I mean, how we want to apply this SynBYSS pitch to combat disease, to reverse disease, informed by gene regulatory network and GRN, the data that I showed earlier on single cell RNA sequencing. We want to come up with a pool high throughput synthetic promoter, which already got a very nice data to detect a specific cardiac cells, actually cell state in cardiac cells, showing that they are fibrotic versus non-fibrotic. And then we want to get into also some microRNA cell-based classifiers, so using the microRNA as a sensors in cardiac function to see if we can detect different stage of disease using those microRNA that are involved in disease. And then hopefully we're combining this platform, we go to the next level. We have some data using this AI. We're trying to apply that AI, like a computing system, to see how we can put a brain in these disease cell types so that they can decide by themselves automatically. So all of this, that the research that we do in the lab, and I hope I can manage, because I just started like 10 months ago recently. So I hope I will be able to apply all this story that I mentioned here for to combat heart disease, gene therapy platform that we are developing here. And at the end, I really would like to say thanks to all my colleagues, ex-colleagues, the amazing, ever-growing community of researchers, the amazing researchers that I have had pleasure working with them and exposed to them, learning from them, from Iran, from Germany, from Austria, now at MIT Synthetic Biology Center. And thank you very much for your attention. And I put my, and I'm happy to take any questions, and I put my email on the slides. If there is any questions, happy to take and answer. And thanks again for the opportunity, Dr. Moon.
Thank you. Great talk.
I hope I was not that fast.
No, it was okay. So let me see whether we have question from the audience. Just give me 1
So
second.
should I stop sharing? It's fine.
And either will be fine. So
Okay.
let me ask, the question in that case, before waiting, while waiting for the question from the audience. So I'm not in your field, so I just wanted to ask some broad or the high level, question. So, there I see some challenges and, opportunity in your field, using synthetic biology. And then, but to go to the holy grail, goal in the future, what would be the remaining challenges and then also opportunity to get there?
So basically, it's very challenging, like what you said. Like actually, synthetic biology and mammalian cells, when it comes to mammalian cells, is super challenging. It's unpredictable, very unpredictable, like when it comes to mammalian synthetic biology. But I mean, at least the good news here is that, I mean, I know stem cell part, I mean, sort of, because this is my experience and my background. So that's why Ron hired me, I guess. But the remaining challenging for all of this, if I want to respond to your question very shortly, is scaling up. The system is not scaled yet, like not in the context of disease, not in the context of cell therapy that I showed earlier, because I had two parts in my presentation. The liver organoid was cell therapy, so we wanted to apply synthetic biology using external molecule and math biology or mathematical modeling to change toward a specific cell type. So that is cell therapy. And then for the second part of my presentation, I talk about applying synthetic biology for disease. None of these approaches are scalable yet. That is the main problem. Specifically for disease one, because the gene regulatory network system is like accessing to the samples. It's not a scale at all. This is number one. Number two, for in the context of cell therapy, synthetic biology to cell therapy is GMP, GLP system, because cell therapy is very expensive, and not everyone have access to GMP and GLP system. So these are all that we know that we are still dealing with that, and that's why we collaborate with industries, with different external collaborators, to make it better and better and better. But yeah, the challenges obviously are there in the context of SynBYSS.
So that's the one challenge
Question or not?
no, that is one challenge I see, but even before thinking about scalability,
Yeah.
I also feel like based on your talk, also based on some other talk I listened to, the other
Uh-huh.
challenge also need to be addressed. The not only, scalability is kind of more likely a little bit later
Down
kind
to
of issue,
translation. Yeah,
but in mammalian cell, in the to implement those kind of technology, we still don't understand how, those kind of cell behave in a
definitely.
predictable way. So the scalability is the next step, I believe. But the first step before going there is something else, like understanding, controlling, gene expression or, whatever, the manipulation. That's not necessarily currently done in real engineering way because we still try an error type of approach rather than just kind of prediction and then work, and that's not the happening right now. So could you elaborate that aspect a little bit more, because that's probably the one thing we need to solve before we thinking about even scale up.
So basically, this is the discussion that we always have with Ron every single day.
Yeah.
That is the system, like biological system, follow analog system or not? They don't. I mean, we don't know actually. So it's just like so unpredictable. We're just trying to make it better and better and better. But the good news is that, like, at least in the context of the work that I'm doing, like synthetic promoter, Ron applied it. He has a paper on that, the specs promoter paper, the synthetic one that I showed in the slides. Actually,
Yeah, I
they
know.
done stem cell and liver organoids, so it's there. But I mean, in the context of disease, that's so challenging
Yeah.
because not just about cells, it's a disease condition as well. You're right. It's so challenging. So we don't know yet. We're just trying to make it better and better, but we are not there 100%. And this is the discussion that we have with Ron every single day. Like, biological system follow analog system, is a biphasic system, is it possible to make it biphasing, toggle switch, bistable? And we don't know. I mean, we're just trying our best to make it closer and closer there, actually. And
Sure.
actually, you tackled the right point, actually, something that we discuss every single day in the lab, to be honest with you.
Yeah, but that's even, person outside that field, mammalian or whatever synthetic biology, I clearly see because I read a lot of papers and I try to read 10 paper a day, only abstract, but so I know the exact problem in the
Yeah,
field, not only
yeah.
my field, but some other field as well. But that is one thing I want to see in near future, but that's not, it's happening, but not happening in a fast enough way I want to see.
Nothing in synthetic biology and mammalian cells are not fast, never.
Yeah,
It's never, it's not, nothing is fast,
I know
obviously.
because I started my one project that using mammalian cell and organoid, but that
Oh, nice.
for almost 4 years, I do not see much progress. So that's I clearly
Well,
know
actually, for liver
how slow.
organoid, at least we were successful to see that effect, not internally, like because what we applied on liver organoid in the context of synthetic biology, it was not internally changing. We didn't manipulate in the cell system internally, like regulatory part. We changed the fate of the cells. We manipulated the cells using external molecules. So we were
Sure.
successful. I didn't show the data because I just wanted to keep track of time, but we have tons of data that we were successful to change this organoid, liver organoid, iPS or liver organoid toward a specific cell type,
Sure.
actually. Webcam progenitor, single cell RNA-seq, PCR, metabolomics. I have tons of that. I just didn't show them. So we were successful for liver organoid, but like what you said, it's very difficult, and we did it externally. When it comes to gene regulation and internally manipulation, that's a totally different level, and it's not easy at all. I know that, and
Sure.
we want to do it in the lab, by the way. So let's see. I mean, if we are successful or not.
Yeah. Also, you said you need to leave earlier than later. So let me, because I don't see any question from audience, so let me close and then we may have a few minute chat or, just leave. Let me close and then we end. Okay. So Thank you all for joining and staying today. We'll meet again on October 18, actually tomorrow, the same time, the same Zoom link. Not the next one is tomorrow due to the rescheduled seminar. Actually, we canceled because of the last minute emergency. One of the speaker last time. We'll have Professor Elisa Franco at UCLA and Professor Justin English at University of Utah. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. And thanks. I stopped recording.
Thank
Just
you.
give me 1 second. Let me see.